All right, in this video we want to discuss another type of cell that's critical to our immune response, and these are the B cells. So as we've discussed all these cells, we'll go through some basics, we'll think about the names, their functions, and then why they're so important for this process. So B cells. So what are some basic facts about B cells that we should recognize? So first, they're a critical component of that immunological third line of defense. Again, this is that adaptive immune response, the one where we learn about specific pathogens and then develop long-term immunity to them. B cells, like all of the cells that are involved in immunology, are white blood cells. So B cells are a type of white blood cell. B cells are a specific kind of white blood cell. They are an agranulocyte. B cells are also called lymphocytes because they're common in the lymphatic system. So you might see them referred to that way. B cells develop in the bone marrow, and we might think that's why they're called B cells, but it's actually not. So B cells were originally discovered when scientists were looking at blood cells in birds. And in birds, the B cells develop in this organ called the bursa of Fabricius. And so they are given the B cell name because of their location in that bursa organ. But anyway, they're B cells. Again, they're a kind of white blood cell, agranulocyte. They're also a lymphocyte. They're developed in the bone marrow. As we wrap up this video, we'll talk about the types of B cells. There's multiple forms of these kinds of cells, and we'll think about what the different forms do. But before we talk about the specific forms of B cells and what they do, let's make this general claim. A B cell's job is to produce antibodies. So before we talk about what B cells do, let's make sure that we fully understand what antibodies do. So what is an antibody? So antibodies are secreted proteins that bind to a specific antigen. So a protein is a small macromolecule made by cells. Any one cell can make millions and millions of proteins. Here's what an antibody protein looks like in a diagram. So an antibody protein has these several parts that are stuck together. They form this Y shape. In general, we'll draw them in this Y shape. What's most important about the antibody is that it has these antigen binding sites at the end. So this antibody protein uses this part of the protein to stick to a specific antigen. Here's another way of representing those antibodies. This is a 3D molecular structure. Again, a little bit of a Y shape, and at the ends out here, there are antigen binding sites. So again, antibodies are secreted proteins that stick to a specific antigen. So I just wanted to show some images that demonstrate the antibodies sticking to an antigen. So here are some antibodies sticking to a specific antigen, which is a spike protein on the outside of a virus. Remember, antibodies stick with the antigen binding site at the end of this structure, and so it's stuck to this virus. Here's another example of an antibody stuck to the B component of this toxin from diphtheria. So this is an AB toxin, and this antibody has identified this B component of the toxin as a specific antigen, and it's stuck to it. Here we've got some antibodies that are specific to a protein in the flagella of a bacteria. So again, the antibodies are stuck to this antigen, some chemical that's in that flagella. So antibodies, they're secreted proteins. So in all three of these, we can see the antibodies are acting independent of any immune cell. We don't see any immune cells here. We only see the antibodies, and they stick to their specific antigen. So we're looking at three different kinds of antibodies. They're each sticking to one specific antigen. So what happens when an antibody sticks to an antigen? Right? So antibody binding to an antigen. What happens? Well, one of two things happens. The first thing is that when an antibody sticks to an antigen, that stimulates immune cells to act on that antigen. So here's an example. Here are some antibodies stuck to the exterior of a bacterial cell, a pathogenic bacterial cell. This macrophage, a professional phagocytic cell, has recognized that the antibodies are stuck to something, and that tells the macrophage that that thing is dangerous and should be destroyed. So this macrophage is in the process of doing phagocytosis on the pathogen so that it can eat and destroy it. So again, antibody binding to an antigen stimulates immune cell function. Here we're stimulating a phagocyte. Here's another example. Here this bacteria, again, is bound by specific antibodies. And this immune cell, this is actually a natural killer cell, is being stimulated to release cytotoxins. Those are just chemicals that are damaging to things that are going to harm the antigen and pathogen that the antibodies are stuck to. So again, when antibodies stick to a antigen, it will stimulate immune cells to perform their function on that thing to protect us. But that's not all that will happen when an antibody sticks to an antigen. The next thing that can happen is that the binding can actually inactivate the antigen. So let's loop back to this example of an antibody sticking to an antigen on this virus. This antigen was a spike protein. We know that viral spike proteins are important for the attachment and entry steps of the viral life cycle. So the virus needs those spike proteins to be able to get into a host cell. Because these antibodies are bound to the spike proteins, they're getting in the way. And now these spike proteins cannot make contact with the surface of a host cell appropriately to gain access. So this virus has been inactivated by the antibodies sticking to it. Another example might be that diphtheria toxin. So these antibodies are stuck to the toxin. This toxin would usually bind to a cell and deliver the A component into the cell and then harm the cell using that A component. But because these antibodies are bound to the B component, this toxin may now be inactive because, again, the antibodies are just kind of getting in the way. So those are the two main things that happen when an antibody sticks to an antigen. Immune cells are activated, and in some cases, the antigen is inactivated. All right. So knowing that B cells produce antibodies and that antibodies have these specific functions, now let's think about how B cells are activated to produce those antibodies. So B cells need to interact with T cells, and specifically helper 2 T cells, to become activated. So helper 2 T cells activate B cells. Without the T cells, B cells do not function. So here's a diagram illustrating an interaction between a helper T cell and a B cell on the left. The helper T cell and the B cell are both bound to the antigen that the T cell recognizes. And in this interaction, the B cell goes through a complex process of identifying an appropriate antibody structure to make that will bind to that antigen. So during this interaction between T cells and B cells learn how to recognize the same antigen as the T cell, and then how to make antibodies specific to that antigen. So we go through this interaction, and at the end, we've got these B cells that know how to make these antibodies that will specifically stick to this antigen that the T cell knew how to recognize. So B cell activation requires T cells, specifically the helper 2 T cells, and after activation, B cells can make these specific antibodies that bind to a specific antigen. So now let's think about the types of B cells that exist after activation. And there's really just two types. We're going to talk about plasma cells. So plasma cells are a specific type of B cell, and memory cells. Again, memory cells are a specific type of B cell. Let's think about what each of them do. So plasma cells. These cells these are cells that produce and secrete antibodies. I sometimes describe plasma cells as antibody factories. These cells are active during exposure to an antigen. So the B cell senses that antigen they know how to make an antibody against is around, and they actively make all those antibodies. They secrete them, they float all around the body, and they bind to those antigens. So here are 3 plasma cells. They've been stimulated by exposure to the antigen, and they're secreting all these antibodies that float away and bind to the antigen, stimulating immune cells and inactivating that antigen. So what about memory cells? If a plasma cell is like an antibody factory, what's a memory cell? A memory cell is a dormant cell waiting for future exposure to the antigen. So sometimes I describe the memory cell as a factory where someone just turned off the power. This factory is not currently making anything, but at any point in the future, if we needed to make that thing again, we could walk in the door, flip the power switch on, and that factory could immediately begin making its product again. So when exposed to an antigen, memory B cells activate into plasma cells. Then the plasma cell actively makes all those antibodies, secretes them, and they float around in the body, protecting us from that antigen. So here's a diagram that demonstrates 3 memory B cells. They've got the antibody component poking out of their membrane, and that will help them be stimulated if they find the antigen in the future. Again, if we stimulate a memory B cell by exposing it to an antigen, it becomes a plasma cell, and it makes millions and millions of these antibodies. All right. So as with all the immune cells, there's a lot of complexity associated with B cells. We're just scratching the surface for this class. The first thing I would add here that you don't need to be aware of for our class is that B cells are actually a special form of an antigen-presenting cell, or APC. So that means that B cells are actually phagocytic in some ways. But again, we're going to gloss over that. We don't need to dive into that complexity for this course. If you learn more about immunology in the future, that might be something you can learn more about. In some cases, B cells can activate without the help of T cells. Again, for our course, this is too much complexity. If you study immunology moving forward, you'll learn all about these processes. But for right now, let's focus on the relationship of T cells and B cells. Finally, B cells go by lots of names. So we're just calling them B cells. We'll call them plasma cells or memory cells based on their function. We're going to ignore all these other names. But maybe if you read more about immunology, you might be exposed to something like a plasmablast, a lymphoplasmacytoid cell, B2 cells, FO B cells, MZ B cells, B1 cells, B-reg cells. Again, these are all forms of B cells that might be important for certain aspects of immunity. But for our class, let's not get lost in those details right now. So we should just really focus on the B cell basics. What are the important details about B cells that we need to be aware of? So first, for our class, they must be activated by T cells. Without interacting with T cells, they will not function. Next, after they've been activated by T cells, they produce antibodies that bind to a specific antigen. We talked about antibody function. After B cells have been trained to make those antibodies, they fall into 2 basic types or functions. Again, there's the plasma cell that's actively making antibodies, or there's the memory cell that's waiting for the future to make antibodies. And I just want to end with this comment about those memory cells. Memory cells, memory B cells, are the key to long-term immunity. I was exposed to chickenpox when I was a child, and the one and only reason that I won't get chickenpox again as an adult is because I have some of these memory B cells that recognize the chickenpox virus and will release anti-chickenpox antibodies if that virus ever shows up in my body again. And that will keep me from getting sick from that virus a second time. So let's end there. As always, if you have any questions, please let me know, and I'll look forward to talking to you all again very soon.